Good evening, everyone, students. I would invite Deepashri ma'am to start the session.
Good evening, everyone. So last class we did the Monera and we completed the Monera part entirely. Remembered? So we completed the entire Monera part and hopefully my screen is visible to everyone. So we did the Monera part, only the bacteria. I have told you that apart from bacteria, there are different things and bacteria is the most important one, and that is why we have read about bacteria. Okay, so hopefully everyone has gone through bacteria and the reactions, the examples, the diseases and everything. And does anyone have any doubt? Anyone, any doubts? Okay. So since no one has no doubt and so let's start with the next topic. So today we will be starting with the spirochetes. What are the spirochetes? Spirochetes are actually something that lives in mud and water. So you have heard the term that don't play in the mud and water for too long because they contains germs. What are those germs? These are actually the spirochetes. Okay, so these are actually the spirochetes who are chemoheterotropic bacteria. What is the chemoheterotropic bacteria? Chemoheterotropic means someone, okay, photoautotropic means someone who can prepare their own food. Like last class I discussed while discussing about the nutrition, I told you about the autotrophic organism. So what is the autotrophic organism? Autotrophic organisms are auto means someone who can do their own task. And chemoheterotropic refers to the organism that requires the organic substrate to obtain the energy and carbon for their growth and development. So in other words, I can say that these organisms, they rely on chemical reactions that includes different types of chemical and organic compounds for their metabolic processes rather than producing their own food through the process of photosynthesis. Like us, we are chemoheterotrophs. We depend on chemicals. Now you will be asking me how we depend on chemicals. Actually, we depend on the plants. We don't eat the vegetables raw. We use different kinds of masalas or the spices. Spices actually act as the enzymes that actually helps in the proper absorption of the nutrients in our body. Okay. Just wait. Just wait for once.
Any issue, ma'am?
Yeah, my screen got fixed somehow. I'm reshaping.
What happened, ma'am?
My screen got hanged.
Okay.
So everything is visible, right? Okay. So sorry for the interruption. My screen got hanged. Okay. So what are these spirochetes? So spirochetes are the organisms that live in the mud and the water, and these are the chemoheterotropic bacteria. So many diseases are caused by them, like Treponema pallidum, which causes syphilis. Leptospira causes jaundice. Borrelia causes relapsing fever. Relapsing fever means recurring fever, the fever that keeps on coming. Spirochetes are found in your teeth. So these are the common examples of the spirochetes, which is another type of bacteria basically. So Rickettsia are the gram-negative, obligate, pleomorphic, but walled intracellular parasite. These are the major characteristics of Rickettsia. in your exam, they may be giving you different characters like gram negative, gram positive, obligate, pleomorphic, and you have to select those options and to tick them. So this slide becomes very important. So rickettsy are gram negative, obligate I have discussed in the last class the difference between obligate and the facultative. Facultatives are strict, but obligates are not strict. But the what is the meaning of the term pleomorphic parasite? Pleomorphic actually, so parasite, we all know the basic definition. Pleomorphic parasite is, pleomorphic means someone who can change their shape during their entire life cycle, like Leishmania, Plasmodium. So these are the pleomorphic they are intermediate between true bacteria and viruses. Chlamydia. These are gram-negative intracellular parasite of about 0.25 micrometer size. What is the difference between intercellular and intracellular? Hopefully, you all have um, learned about learned about intracellular and intercellular competitions. Intercellular competition inter-school competition means your school is competing with another school. And intra-school competition means the competition is taking part among the students of your own school. So that is intra-school and inter-school. Inter means with different school, intra means within themselves. So chlamydia are the gram-negative intracellular, means something that resides within the own cells. So intracellular parasites of about 0.25 micrometer in size, grouped along with rickettsy, but different from the rickettsy in the form in actually reproductive cycle. Okay, they consist of reticulate bodies and the elementary bodies inside host phagocyte. These are the different structures of chlamydia that actually help them to infect. So chlamydia trachomatis causes conjunctivitis, sexually transmitted non-gonorrheal urethritis, epididymitis, cervicitis, proctis, and lymphogranuloma venereulum. So these are the different disease, and these are the different disease which are actually caused by chlamydia trachomatis. Chlamydia trachomatis is an important organism under chlamydia, under the genera chlamydia. So these, and what are the diseases caused? These are the important disease, and most common is the conjunctivitis. Maybe in our entire life, we all have been infected with chlamydia trachomatis causing conjunctivitis at least once. Okay. So chlamydia pneumoniae causes pneumonia and bronchopneumonia. This is another species of chlamydia. And next comes the actinomycetes. So what are actinomycetes? These are mycelial bacteria, which form radiating colonies in structure. Radiating colonies means, mycelial means myceli can be, mycelium is actually like plural form of the mycelium is the myceli. It is actually the vegetative part of the fungus that consists of different branches, and these branches may be interconnected with each other. Okay, so this you can say as the root system of the fungus, which is like the root system of the plants. Root system of the fungus is the mycelium. So mycelium grows by hyphae, which actually breaks down and absorb the nutrient from the surrounding. So this is the basic structure for
Ma'am, what is the actual meaning of actinomyces?
actinomycetes is a type of bacteria. Bacteria have different classes, you know. So actinomycetes is actually a class. Though I don't know what the actual term actinomycetes means, but it is actually a class or you can say a division for bacteria. These are all bacteria, okay? And radiating colonies means when if you are studying microbiology, you will understand that colonies for each bacteria varies. Some form round colonies, some form star-shaped colonies, and actinophycetes forms the radial type radiating colonies. And these are actually shown by not only actinomycetes but any fungi, yeast, they show radiating colonies in the culture. Okay. So because of these, actinomycetes are called ray fungi. So very important question. They can frequently ask you that what is ray fungi? So actinomycetes is actually called the ray fungi. Mycelial form is reduced in Mycobacterium and Corynebacterium. These are different. See, we are saying this is Mycobacterium and Corynebacterium. We are using the term bacterium, but they are actually the parts of actinomycetes. So mycelial form is reduced where? In Mycobacterium and Corynebacterium. These small points you have to remember. And mycelia are very small. They are like in a diameter of 1 millimeter or less. And actinomycetes wall, it is the walls which is being written here. The walls are actually not the walls of the bacteria. It's saying the walls of the actinomycetes. They contain mycolic acid, which is the fatty acid, lipids, and waxes. Okay, so there is just lot to remember. So is this part clear? Is this part clear, everyone?
Yes, ma'am.
Just okay. So if I ask you from this part, that we have learned about spirochetes, chlamydia, and actinomycetes. So what are these? What are these? These are bacteria or the viruses. What are we actually learning? We are learning the subdivisions of different types of bacteria. Till last class, we learned about the basic structures of bacteria, right? What is the basic bacteria? Their growth, their their nutrition, their growth media, and so on and so forth. But now we are learning specifically about the specific types of bacteria that frequently comes in your question. Okay, something more I want to say, these parts may not be present in your NCRT, I know. But just because Neet always don't give you questions directly from NCRT. Extra parts are also given. That is why I have included certain things that you need to remember. Okay. So there are different modes of the reproduction, obviously, because bacteria, you have seen how many different types of reproduction are there. Binary fission and budding. And the actinomycetes being a part is also a different type. So different modes of reproduction are by conidia, sporangiospore, arthrospore, oidia, and fragmentation. So most of the actinomycetes are saprotrophic. Can anyone tell me what is the definition for saprotrophic, or does anyone have any idea what is saprotrophic? Anyone? No one? No one wants to try also? Just anyone, just say that what is actually saprotrophic.
Ma'am, can you repeat the question again?
Yeah, I told you, can anyone tell me what is the meaning of saprotrophic, the term saprotrophic? What mode of nutrition is saprotrophic? No one? Actually, you all know this. Saprotrophic means when the organism is dead and the other organisms are obtaining their nutrients by decomposing and breaking down the dead organic matters. So that is the saprotrophic mode of the nutrition. Okay, like the fungi, bacteria, protozoa, detrivores. Okay, so these all are saprotropics. So saprotropic and constitute an important composer of decomposers like actinomyces, streptomyces. So a few are pathogenic in plants, animals, humans like Mycobacterium. Mycobacterium tuberculosis. Mycobacterium tuberculosis causes the disease tuberculosis. So this is actually harmful. So this is caused by that organism. Okay. So in pathogenic actinomycetes or mycobacterium, a derivative of mycolic acid called mycoside or cot factor is involved in causing the disease. A number of the antibiotics are produced by actinomycetes like the streptomycin, chloramphenicol, tetracycline, tetramycin, terramycin, erythromycin, biomycin, novomycin, nystatin. Okay. So what are these? These are the different name of the antibiotics which are actually produced by whom? Produced by the genus of the actinomycetes. Okay, so the actinomycetes and streptomyces is a part of the actinomycetes. So mycobacterium can cause the disease, but streptomyces is actually useful. Streptomyces doesn't cause the disease. Streptomyces instead produces different antibiotics, and antibiotics, we all know that antibiotics are something that actually helps the organism to kill other types of the bacteria. Okay, so among this, nystatin is very important because nystatin, this nystatin is an antifungal antibiotic. Okay, this is an antifungal antibiotic. So if you are infected with any fungal kind of disease, they can be infecting you with, they can be treating you with nystatin. So who was the first person? Okay, just remember, first antibiotic was the penicillin. But the terminology, antibiotic, who gave this term? Waxman gave this term. A.S. Waxman, Abraham Selman Waxman gave this term. Who also extracted the first antibiotic from Streptomyces griseus and named it as Streptomyces. For this, Waxman was given the Nobel Prize in 1952. So you from this, you came to know that streptomycin is actually obtained from Streptomyces griseus. And for this, Waxman was given the Nobel Prize in the year 1952. So, archaebacteria. So this is a very primitive type of the organism. Okay, so the first cell, archaebacteria and the eukaryota. Archaea and the eubacteria. So archaea is again divided into archaebacteria and the eukaryota. So archaebacteria is one of the most primitive type of the organism that has evolved, and now they are just fossils. Okay, so archaebacteria is considered to be one of the most primitive, like you can say them, they are the first cells of the planet. So a group of the most primitive prokaryotes, which are believed to have evolved immediately after the evolution of first life. Remember, first, first cell is not the archaebacteria. But after the first cell, something evolved, and that is the archaebacteria and eukaryota. Okay, so is this part clear? Archaebacteria is not the exactly the first cell. When you all will be learning the evolution, then you will understand that that's not what the first cell is. So first cell, then after evolution, that's the archaebacteria. So now they are just living fossils. So archaebacteria are considered and characterized by the presence of peptidoglycan in their cell wall. Okay, this is important. So they have peptidoglycan, but this peptidoglycan is pseudopeptidoglycan. Okay, so don't write the absence, don't use the term absence. because the term absence is not totally wrong, but it should not be said absence. So you instead you can say that it is actually made of the pseudopeptidoglycan. What it is made up of? Pseudopeptidoglycan. Just note that when you will be learning, you can read this. So it has pseudopeptidoglycan. Okay, all remember this. So it has pseudopeptidoglycan. Okay. So instead the wall contains proteins and non-cellulosic polysaccharide, it has pseudomurine. See, I have told you that murine is the another name for the peptidoglycan, if you remember. So this is the pseudomurine. So pseudomurine and the pseudopeptidoglycan are same. And they are present in methanogens. Methanogen, remember, methanogens are the types of the archaebacteria. Okay, so if I say this is the first cell, okay, then from these two things develop. One is the archaea, and this is the eukaryota. Sorry, this is the eubacteria. Okay, archaea and eubacteria. Now from this archaea, here comes the two things. One is the archaebacteria and another is the eukaryota. Okay, and from this eubacteria here comes the photosynthetic and the non-photosynthetic bacteria. Is this part clear or should I repeat? Did everyone note it down? Did everyone note this down? What I have said just now. Please, students, please be responsive. Did everyone write this? This is the first cell, this is the archaea, and this is the eubacteria. So archaea, and this is the archaebacteria, and this is the eukaryota. Okay, so this is the entire classification for the life. And their cell membranes are characterized by the presence of branch chain lipids. Remember, for bacteria, that is for prokaryotes, and for the eukaryotes like us, we have unbranched lipids. Unbranched lipids. Only archaebacteria has this branched lipid. Okay, remember this always. Now, archaebacteria even live under extreme hostile conditions where there are very few other organisms like salt pans, salt marshes, hot spring. They have this ability. And their RNA are quite different from their other organisms. Obviously, that is the reason they can tolerate extreme conditions. And archaebacteria are of two broad categories: obligate anaerobe and the facultative anaerobes. And archaebacteria are methanogen, halophiles, and the thermophiles. Methanogen means one who can live in extreme methane containing situation. Thermophile means one who can live in extreme temperatures, like either too cold or too hot. And halophiles mean one who can live in higher salt concentration. Okay, so just remember that normally we say that bacteria don't live under extreme cold or extreme hot weather, but this archaebacteria has the ability. Just use your common sense. Initially, our earth was too hot for the living organisms, right? Slowly, the living organisms started evolving. And then thermoacidophils came. These are the archaebacteria. Why? Because at that time, temperature was so high that these organisms came and started surviving. And then from that organism, other organisms started living. So archaebacteria are the living fossils because they represent one of the earliest form of the life which has been experimented. and on the absorption of the solar radiations for the first time. They lived comfortably under anaerobic conditions and developed techniques to oxidize the chemicals in the substratum on the availability of oxygen. What I told you, from the archaebacteria, then the other higher organisms started coming into life. So when we will be learning the evolution, we will be dealing this part in the extreme detail. But now, for now, you have to understand that they lived in the anaerobic condition, that is without oxygen. They were thermophiles because they can live in extreme hot conditions. And they also try to live and oxidize the chemicals. Why? Because when they oxidize the chemicals, they in the chemicals which is present in the substratum, on the dependence of the oxygen, from there different other organisms started evolving. See, we are chemoheterotrophs. We oxidize chemicals mostly depending on the availability of oxygen. So this shows that our great super great ancestor was an archaebacteria. Okay, so these archaebacteria are strict anaerobes. Nutritionally, they are autotrophs because they obtain both energy and carbon from decomposition products. They can prepare their own food. Remember, from them, chemoheterotrophs developed, but they themselves are the autotrophs. Remember, so they can occur in marshy areas where they convert formic acid and carbon dioxide into methane. And this capability is commercially exploited in the production of methane and fuel gas inside gobar gas plants, like methanobacterium, methanococcus. So that is, they can produce this methane gas, and you know methane gas can be used as a fuel also. So for that, they are used in gobar gas plants, and these are the methanobacterium, methanococcus, and so on. Some of the methanogen archaebacteria lives as the symbiont. So inside the rumen, cow, buffalo, and help the rumens in the fermentation of different kinds of cellulose. These bacteria are not present in our gut. And that is the reason, unlike cows, we can't digest all the plants. We have to cook them before eating them. Halophiles are named because these can live in the rich salt concentration, like salt pans, salt beds, salt marshes, halobacterium, halococcus. So they are aerobic chemoheterotrophs. Their cell membrane have red carotenoid pigments for the protection against harmful solar radiation. And under anaerobic conditions, halophiles cannot use the external material. Okay, so they can survive in extreme salt condition, even in your pickles also. The pickle, that is the salty thing that we eat. So in that pickle also, the halophiles survives, like salt pans, salt beds, salt marshes, and so. So at this time, they subsist on ATP synthesized by membrane pigmented system for the from solar radiation. Halophiles growing in the salt pans and salt beds give offensive smell and undesirable pigmentation to the salt. You have never seen this because this area is not suitable for the halophiles to grow. They grow in extreme salt, like more salt of a salt, like father of the salt, salt marshes, salt beds. That is only salt and salt. So their halophiles can grow and give too bad smell when they are growing. And also the salt becomes undesirably pigmented. So halophiles are able to live under high salt conditions due to the following reasons. Presence of the special lipids. I told you they have branched lipids unlike us, so they can live in extreme salt condition. Occurrence of mucilage covering. This is just a covering. Absence of the sap vacuoles and hence plasmolysis. Plasmolysis means they don't get if the water. Okay, so common thing is you are thinking that if there is a system, suppose there is a system and this system has few salts, okay, and in the surrounding this system has a lot of salts, like this part has the lot of salts, Huge amount of salts are also present here as compared to this. So here salt is less means here water is high. Here salt is more means here water is high. Water is less. So water will tend to flow from here to here, and this will get plasmolized, and obviously this region will die. So you can think that if this is the bacteria and they are residing in the extreme salt condition, so the water from their body can also pass from here to here and they can eventually die. But no, they don't have any sap vacuoles, they don't have any water in their body only, and hence this plasmolysis can never occur. So they have high internal salt content also. So is this story clear what I have told? The concept of the sap vacuole and the plasmolysis. Is this part clear?
Yes, ma'am.
Okay, good. So now comes the thermophiles. What are thermophiles? That live in the extreme temperatures and extreme pHs. Like archaebacteria have dual ability to tolerate high temperature as well as high acidity due to two reasons: branch chain lipids. So that is the reason I told you to remember this branch chain lipids, because branch chain is something they have to have. So branch chain lipids in the cell membrane and the presence of special resistant enzymes capable of operating under acidic conditions. So they live in hot sulfur spring where the temperature may be as high as 80 degree. pH can be low as 2, like thermoplasma, thermoproteus, and so on. So they are tolerate high temperature, okay, and high acidity as well. So they have branch chain lipids in the cell membrane and also the presence of specific resistant enzymes which are actually capable of operating them under acidic conditions. So that is the reason they live in hot sulfur, hot springs, extremely acidic conditions, and so on. Basically, these are the basic reactions you again have to remember, that they oxidize the sulfur to produce sulfuric acid H2SO4. Okay, that will be H2SO4 and not H2SO2. So this will be H2SO4. So these are chemosynthetic, obtain energy for synthesis of food from oxidizing sulfur. Okay, thermophiles are chemosynthetic, but archaebacteria are what? They are the autotrophs. Remember this. So if the conditions are anaerobic, thermoacidophiles may reduce sulfur to H2S. This entire reaction happens only in the anaerobic condition. And bicarbonates are also precipitated into the carbonate form by their activity. This is another example. Okay, so mycoplasma, we have just learned what is a mycoplasma. These are smallest, okay, smallest, simplest prokaryote, free living prokaryote. And they were discovered in the cattle who were suffering from pleuropneumonia by Nocard and Rocks in 1898. And these organisms are often called MLO, that is mycoplasma-like organism, or PPLO. And their size ranges from 0.1 to 0.15 millimeter. So this is, they are in the millimeter range, and they are the smallest. Remember this, smallest and the simplest free living prokaryote. So PPLO, that is the pneumonia-like organism, and they were isolated from where pleural fluid of the cattle. Who discovered? Nocard and Rocks. Okay. So plasma membrane forms the outer boundary of the cell. Simple. We know this. And a substantial amount of polysaccharide having an acetyl glucosamine are associated with the membrane, which is rich in cholesterol. This is for PPLO. Okay, this is for mycoplasma plasma membrane. Their plasma membrane contains polysaccharide that has an acetyl glucosamine along with the cholesterol. Remember this. that the archibacteria have branched chain. But this plasma membrane have the polysaccharide that has an acetyl glucosamine, which is rich in cholesterol. They don't have any cell wall. Very important line. They don't have any cell wall. This is very important line, and often a lot of questions have been asked from this statement, as their cell wall is absent. And due to the absence of the cell wall, they can change their shape, and that is why they are pleomorphic. This is common sense. See, if a shape is present, then you can't change the content present inside it to anything you like. But water has no boundary. You can keep the water like in a container like this. You can keep water in a container like this. You can keep water in a container like this bat shape. So whatever, water can change their shape. Similarly, this mycoplasma have no cell walls, and that is why they can easily change their shape, and that is why they are quite pathogenic, because sometimes the bacteria can't enter inside our body due to their shape, but now they can enter inside our body. So like other prokaryotes, mycoplasma possess one envelope system. Okay. They lack the organized nucleus, endoplasmic reticulum, plastis, mitochondria, Golgi bodies, lysosomes, centriole, flagella, etc. These are the things they lack. I have included this because last year this question came, that what they lack, and these are the things they lack: nucleus, endoplasmic reticulum, plastid, mitochondria, Golgi body, lysosome, centriole, flagella. And the genetic material is represented by a single DNA duplex, which is naked because of the absence of the histone association. DNA duplex is not compacted as in other prokaryotes, but instead lies coiled throughout the cytoplasm. Ribosomes are 70S and granules of various types occur here and thereby in the cytoplasm. Mesosomes are also absent. Okay, so these are the basic things you have to remember about mycoplasma, that their DNA is duplex, not very compacted. It is in the coil form in the cytoplasm. Ribosomes are 70S. You don't have to remember this because you already know that it is a prokaryote. So obviously ribosome will be 70S. And enzymes lie freely both in the cytoplasm as well as associated with that of the plasma membrane. DNA possesses a replicating disk at one end. Okay, so when the cell divides, so from one cell, it becomes two cell like this. Okay, so this is the mother cell and this is the two daughter cell. So the DNA from here needs to be copied so that this daughter cell and this daughter cell both can have the DNA. And that is why the DNA possesses a replicating disk. And that is why this replication and separation of the genetic material is important. And this replication disk actually helps to copy this DNA. Like it is a Xerox machine for the DNA. Okay, that actually copies this DNA and send to this and this. Some of them live as saprophytes, but majorly parasitic plants and animals. The parasitic habit is due to inability of most mycoplasma to synthesize the required growth factors like Mycoplasma gallisepticum. Okay, is this part clear? Does anyone have any doubt? Anything? Okay, so you will know that in our body, ATP is required. ATP is required for different types of the functions. Like ATP is needed for for you even to walk also you need the ATP in your body. But it is not always possible to get this ATP. Body has to make this ATP. So for making this ATP, where do they go? They rely mainly upon the electron transport system. So electron transport system is not present in Mycobacterium for any reason. Okay. They are absent in Mycobacterium. Okay. And reproduction occurs mainly by fission or the budding yeast. or first forming a branching filament with numerous nuclear bodies, followed by constriction in between the nuclear bodies and the separation of the cells as the new individual. And they mostly produce the pleuropneumonia in domestic animals, and mycoplasmal urethritis in human, little leaf disease of brinjal, and witch's broom in plant. So see, these are the organisms who are causing disease in every spectrum of life, like electrons transport system is absent, but they can be affecting the organism of all domains like humans, plants, and even animals also. Okay, so that's how much pathogenic they are. Why? Because they don't have this cell wall, and that is why they can change their shape to like literally anything, and that is why they can enter anybody, and thus they can also create the disease. Mycoplasma are not being affected by penicillin, inhibitor of the wall formation. Both are being but are inhibited by tetracycline. Actually, penicillin targets the cell wall, but mycoplasma has no cell wall, so it is not being affected by penicillin. Rather, it is being affected by tetracyclines. So, next comes the cyanobacteria. So, cyanobacteria is another type of the bacteria. These are gram-negative, performs oxygenic photosynthesis like the plants. And these were the first organism that makes the atmosphere aerobic. And thus the blue-green algae live virtually in all the environments that contain the water. Organisms range from tropics to plains, and they occur in soil, freshwater, ocean, and in lakes and in the ocean, they form part of the planktons. So what are these cyanobacteria? These are again types of the bacteria which are gram-negative prokaryotes, and they perform oxygenic photosynthesis like plants. So these are autotrophs because they can perform photosynthesis, and these were the first organisms to make the atmosphere aerobic. Okay, because they perform oxygenic photosynthesis, they release oxygen, and when they release oxygen, this oxygen is being transported. Where? This being transported to the atmosphere that actually makes the environment aerobic. And the blue-green algae lives virtually in all the environments that contain water. So these actually gave rise to the modern day environment, making the atmosphere aerobic. So they were the first organisms to make the atmosphere aerobic. And these organisms range from tropics to plains, as they occur in soil, freshwater, ocean, and in lakes also, and in the ocean also. They form the part of planktons. Okay, like zooplankton, phytoplankton, and so on. So what are the general characteristics? Some cyanobacteria live in the icy water of the glaciers and others in the hot spring where temperatures reach 85 degrees Celsius or more. So cyanobacteria are kind of thermophiles. They also live in extreme temperatures. Here also they live in extreme temperatures, but they can tolerate extreme acidic environments, but cyanobacteria can't. Okay, so some blue-green algae as symbionts with the other organisms like lichen. This is very important because they ask the association. Blue-green algae in symbiotic association with whom? Lichen. Okay, so cyanobacteria can be unicellular, which is the spirulina, colonial, which is the nostoc, filamentous, which is the oscillatoria. So filaments contains one or more trichome, which is inside a mucilage sheath. Okay, so some cyanobacteria live in the icy water, like in the glaciers. Some live in the extreme temperatures, and they are in symbiosis with the other organisms like lichen, and they contain one or more trichomes inside a mucilage sheath. So, continuing with the general characteristics. Trichomes may be homocystous. So homocystous means those who have no cyst-like structures. Okay. So generally, cyst-like structures are present, but cyanobacteria will have no cyst. Always remember this, like Oscillatoria. Then heterocystus means someone with cyst, like nostoc and the anabaina. Nostoc anabaina are actually the nitrogen fixing bacteria. They live in azolla, Atroceus, Cycas, Gunera, and nodules of the Trifolium alexandrinum. So these are the plants, they make the nodules and they fix the atmospheric nitrogen there. So oscillatoria shows the apical oscillations, or heterocystus, like the with heterocysts, like nostoc, anabina, and so on. The cell wall possesses an outer sheath which is jelly-like, slimy, and the mucilaginous. And cell contents are divided into two regions: outer chromatoplasm, which has the photosynthetic pigments in free thylakoids and inner colorless centroplasm, and cell wall is two-layered, whose inner wall is made up of peptidoglycan or mucopeptides, which are built from amino sugars like glucosamine, and amino sugars may be muramic acid, diamino pamelic acid, and so on. So these are the special type of the cell wall. They have peptidoglycan. Archaebacteria have no peptidoglycan. But these cyanobacteria have peptidoglycan or mucopeptides. And amino sugars are also present, like glucosamine, muramic acid, diaminopimelic acid, and so on. So these are the specific characteristics of the genus Cyanobacteria. Okay. So in the cytoplasm, actually photosynthetic lamellae are present. These lamellae actually are the region where phycoerythrin, allophycocene present, plus chlorophyll. And I've told you cyanobacteria can perform their own photosynthesis. Very important characteristics. They were the first. They originated before plants. So they were the first who made the environment oxygenic. And that is the reason the lamella of the thylakoids contain dominating pigments like phycocyanin, allophycocyanin, phycoerythrin, in addition to chlorophyll. So bacteria and blue-green algae lack the mitochondria through vacuoles, endoplasmic reticulum. Sterols are absent in bacteria as well as the cyanobacteria also. So these are the general characteristics or the basic characteristics which the cyanobacteria actually has, that actually help them to perform photosynthesis. Remember, they don't have any mitochondria or a vacuole or endoplasmic reticulum. They just have the thylakoids, which is the plastid-like structures. And actually, these thylakoids contain phycocyanin, allophycocyanin, phycoerythrin, in addition to chlorophyll A, that actually helps them to perform the process of photosynthesis. Okay. Next, true nucleus is absent, and that is why they are prokaryotes and their DNA is just like freely, they are just swimming in the cytoplasm, if that's the correct term to use. So two fibrils of DNA are either distributed throughout the cell or concentrated in the central part. It's the chromosome resembles the bacterial chromosome. Gas vacuoles are often present, which actually regulate the buoyancy in water, and the color changes from green to deep purple to blue-green. And these colors are produced by different proportions of different pigments like chlorophyll A, carotene, xanthophyll, blue cyan, phycocyanin, red phycoerythrin, and so on. So this color changes from green to deep purple and often blue-green. And these colors are produced by different proportions of different pigments like chlorophyll A, carotene, xanthophyll, and so on. Thylakoid membrane contains chlorophyll, carotene, xanthophyll. Attached to them are phycobilisomes, and they have three types of water-soluble pigments: phycocyanin, allophycocyanin, phycoerythrin. Okay, so these are the different pigments, accessory pigments. Actually, for the process of photosynthesis, not only chlorophyll is required, but other pigments are also present that increase the level of the photosynthesis. So phycocyanin, allophycocyanin, phycoerythrin, these pigments are present. And what it is being written here in bracket, phycocyanin and allophycocyanin, both are blue. Like these pigments are blue in color. And phycoerythrin, these are red in color. Okay, so these are actually red in color, and phycocyanin, allophycocyanin, both are blue in color. They actually have accessory pigments that help them to get the sunlight and to perform the process of photosynthesis on their own. Okay. Many forms show the Gaiduka phenomena or the chromatic adaptation, where color changes according to the wavelength of the light. Actually, light has different wavelengths. Okay, all the wavelengths are not being used by us or used by all the organisms, but there are different kinds of the light. Like Gaiduka phenomena states that cyanobacteria in different wavelength of light can be of different color. They can be asking you question directly from what is Gaiduka phenomena. Okay, so remember it is the chromatic adaptation where they can change their color due to different wavelength of light. Trichodesmium erythraeum is a reddish colored cyanobacterium which occurs in such abundance in sea after its color, and which is the Red Sea. This is very important. This question has come also, that Trichodesmium erythraeum, they have asked the name of the organism, that why Red Sea is named red, because of the presence of Trichodesmium erythraeum. Okay, so it's a reddish colored cyanobacterium that occurs in abundance in sea, and thus it is named as the Red Sea. Nucleoid is in contact with a group of the coiled membranes called lamellosome, and lamellosome develops from plasmalemma. Plasmids or the additional small DNA rings may occur, which is the 70s ribosomes are abundant. Cell inclusions like alpha granule, beta granule, volutin granule, and polyhedral bodies are even present there. So nucleoid is what? Nucleoid comes in a contact with the coiled membranes called lamellosome. Lamellosome develops from plasmalemma, and additionally DNA rings are also present, like 70s ribosomes are abundant, and cell inclusions like alpha granule, beta granule, these are actually present with the volatile, these cell inclusions, and these are that they like store the food in them. Cyanobacterial starch like glycogen, beta granule is used to store the lipid droplets. Okay, so these are the major characteristics. Okay, I will be stopping here up to this slide. Then you can share me your doubts. Okay, the reserve food is cyanophycine, mixophycine starch, and cyanophycine and mixophycine. Cyanophycine, another name is mixophycine. So that is actually a form of starch for cyanobacteria. And flagellas are absent in the vegetative as well as the reproductive phase. Well-marked reproductive organs, sexual reproduction, motile reproductive bodies are also absent in cyanobacteria, because they reproduce by conjugation, transformation, transduction. Last class, if you have studied, you will remember that we discussed about conjugation, transformation, transduction. So reproduction is very simple and takes place by vegetative means only. No trace of sexuality has been ever recorded, but gene recombination can occur through conjugation, transformation, and transduction. Okay. So is this part up to this? Is it clear? I know this chapter involves a lot of memory tasks, but I have tried to give you in point basis so that it becomes easy. After each and every class, if you are memorizing, like after within 1 week, if you are memorizing each and every characteristics, hopefully that will be easier for you to remember. Okay. So, ma'am, this is just from my end. Does anyone has any doubt? Anything you want to ask me? Anything?
Yes, students, you can ask doubts if you have any. Hope they are clear, ma'am.
So no one has no doubt. No one? The modes of the reproduction, the types of the nutrition, everything, every term is clear to all of you. Because if this term is clear to you, even if in the question paper something different types come, you can use the logic and answer. So I'm just stressing over you. Every point, every term is clear to you or not. Most of the things are already written in the PPT itself. So apart from this, any term, anything which you have, if you are having any confusion, you can ask me. So is this part clear to everyone?
Hello, ma'am.
Yeah, say.
Ma'am, what is difference between branch chain lipids and unbranched chain lipids?
Branch chain lipids means when you will be learning the lipid structure, you will be seeing some lipids are straight chains only. Okay. But some lipids have branch chain. Means there is the main chain, along with it, there are different other chains. And it looks like a tree. Okay, so those branch chain lipids, those are the primitive forms of the lipid. Those are present only in the archaebacteria, and those actually helps the organism to survive to specific conditions like high temperature, high pressure, and so on. Whatever we have learned in today's class, abnormal conditions, whatever they are living. Okay.
Okay, ma'am. Thank you.
Anyone has any doubt other than this? So, okay. Thank you. Thank you for the patience. Over to you, ma'am.
Yes, ma'am. Thank you, ma'am. Yes, students, tomorrow you have a mock test. Sorry, weekend test. Okay. So tomorrow we will send the link after 2 in the group. You can attempt and write the weekend test. And again, we have class from Monday morning 5 to 7. Okay. Uh, Umar, can you please unmute?
Yes,